we're going to discuss the reverse transcriptase polymerase chain reaction, or popularly known as your RT-PCR. This is the second part of our lecture about amplification techniques. Initially, we discussed PCR and now RT-PCR. Today, we're going to learn the difference between the two, the function of RT-PCR, and the current uses in our clinical diagnostic environment. Okay, so let's proceed. So what is PCR and RT-PCR? Basically, RT-PCR is an offshoot of your PCR, a modification. Instead of a DNA as a starting material in PCR, in RT-PCR, we use RNA as a starting material to create the amplicon that we desire. No? Please take note though that the amplicon produced in RT-PCR is DNA as well after converting this to the RNA to a DNA. So it's the same product like that of your PCR, DNA. However, the starting materials are different. So RT-PCR is used today in many clinical diagnosis and quantification of RNA virus infections like HIV, hepatitis C virus, and of course the coronavirus that we have right now, a big problem, the SARS-CoV-2. We detect the RNA of that, and other many retroviruses. So in this slide, I'm showing you the basic principle of RT-PCR. Okay, so in RT-PCR, the RNA population is again converted by reverse transcription, and then the cDNA is amplified by the polymerase chain reaction. So that's basically the whole process. First, we convert the cDNA, we convert the RNA to become a cDNA by reverse transcription, and then the cDNA by amplification by polymerase chain reaction. The cDNA amplification step provides opportunities to further study the original RNA species, even when they are very limited in amount or abundance. The common applications of RT-PCR include detection of expressed genes, examination of transcript variants, generation of cDNA templates for cloning, and sequencing. So let's have a brief overview of this one. So initially, this one, the blue bar, it represents your RNA. And usually an RNA, this one, in this diagram, it consists of an AUG, start codon, and a poly-A tail. So initially, a primer will attach to the poly-A tail of the RNA, and this will be acted upon by your reverse transcriptase to continue adding your DNTPs. Okay, as seen in the first, then in the third, and in the fourth one. So after completing the process by the reverse transcriptase, an RNA hybrid is formed. This is the RNA and the complementary DNA. So to remove the RNA, there is what you call kasi RNase H activity in reverse transcriptase. And this is able to remove the initial RNA template from the complementary DNA. And therefore, when this is done, you have now your solitary complementary DNA, and then this will have it will become a double-stranded DNA, and then it's ready for the amplification steps that we follow in the initial PCR. So that is actually the basic principle of this RT-PCR. Okay, so again, in RT-PCR, we use RNA as template instead of DNA for the nucleic acid amplification. Okay, and RNA is first transcribed into double-stranded DNA, which is then amplified through the PCR. Okay, so the RNA becomes, through the action of reverse transcriptase, become eventually a DNA, and it needs a primer and a reverse transcriptase for this to happen. So, in summary, RT-PCR starts by synthesizing DNA from isolated RNA sample, then degrading the RNA proportion of the hybrid through its RNase H function of the reverse transcriptase and synthesizing a double-stranded DNA. Reverse transcriptases in RT-PCR are thermostable and also generate cDNA with high processivity after the primer anneals to the RNA template. Next, amplification proceeds in the usual PCR procedure. However, the end product, the amplicon, remains to be a complementary DNA. Okay? So, here, we're gonna discuss the important elements of an RT-PCR. No? So, these are the components that we know, no, as we've discussed in the previous slides. We have the reverse transcriptase here, and then the reverse transcription primer, magnesium, and all of the necessary components for PCR, the PCR portion of the RT-PCR. Again, the reverse transcriptase is an RNA-dependent DNA polymerase, and it has three functions. No, it synthesizes a cDNA strand on an RNA template, remove the RNA strand from the DNA-RNA duplex or hybrid that we call, and then synthesize a second DNA strand on the DNA template. Okay, that's very important to know. The reverse transcription primer is a single short DNA molecule that acts as starting points for the reverse transcriptase, since reverse transcriptase cannot initiate DNA synthesis without a primer. Magnesium serves as cofactor in reverse transcription reaction, and sometimes magnesium can be replaced with manganese. And of course, in the previous lecture that we discussed, all of the necessary PCR components for the PCR portion of RT-PCR. Components of your RT-PCRs, and there are various types available for the RT-PCR. The first are your oligo DT primers, and these are usually 12 to 16 BP long and contain repeated deoxythymines in their 3' end that bind to the poly A tail at the 3' end of the DNA strand. These primers proceed to amplify in the 5' end direction, similarly to gene-specific primers. When a long amplicon or full-length transcript is desired, either gene-specific or oligo-DT primers are favored types of primers used. No, oligo cannot be used to transcribe RNA lacking polyamino tails because they need to attach to these degraded RNA or RNA that has considerable secondary structures. No, so that's the important considerations for these types of primers. The next are random primers, or what they call hexanucleotide. No, so these primers are used when nothing is known about the RNA template, or in case where there's substantial amount of RNA secondary structure in the specimen. No, these will anneal to mRNA at any complementary site. So these random primers bind themselves less specifically to the template with multiple primers attached to one strand. Then amplification progresses towards a prime end from each primer, terminating whenever the point of replication from another primer is reached. Random primers work well again on degraded samples of RNA. So, to excise and replicate just a portion of a longer transcript, random oligo primer or oligo-DT with a random monomer will produce cDNA for portions of a full-length transcription. So that's it. The disadvantage is that cDNA is made from all RNAs and that are sometimes not desirable and can dilute the mRNA signal. So the third type are specific oligonucleotide primers or specific sequences primers. So at these kinds of primers attach only to the region of the template's genetic code complementary to their own and begin replicating at the 5' end direction. And these types of primer are usually the ones that we use in what you call a one-step RT-PCR that I will show to you later. So those are the what you call a specific initiation point to known sequences, and very limited on one gene of interest. Okay, so those are the various types of primer. So earlier I was telling you about the two types of methods in RT-PCR, the one-step and the two-step RT-PCR. So what is the difference between the two? No, RT-PCR and PCR can be done in any of those ways. No, so please, before we discuss that further, please take note of the following. No, this one in the pictures are the elements, no, of the RT-PCR. These are the test tubes. No, they represent like that. Okay, this is the image for the one-step PCR, and this is for the two-step PCR. Okay, so in one-step PCR, the reagents of the RT and the PCR reaction, no, the reverse transcriptase PCR and the PCR, are added to a single tube, allowing both reactions to occur simultaneously. No, so you just add this and then in the tube, and then all of them will undergo the procedure, and then you will have your product, the amplified cDNA. This assay combines the reverse transcription in a single tube and buffer, as the name implies. So this uses a reverse transcriptase along with a DNA polymerase. The RT-qPCR only utilizes sequence-specific primers, as mentioned before. This is for assessing many RNA samples and high-throughput applications. This is the best method when only a few assays are run repeatedly. Okay, so those are the uses. In two-step PCR, the RT reaction, the reverse transcription reaction, occurs separately from the PCR reaction. That's why it's called the two-step PCR. No? So in this assay, the reverse transcription and PCR steps are performed in separate tubes shown in the figure, that you can see, and with different optimized buffers, reacting conditions, and priming strategies. So several primers can be used, which are the oligo-TT primers, random hexamer primers, gene-specific primers, or a combination of them. And this is best for assessing multiple targets from few RNA samples. Okay, so those are the two things. And then so aside from those uses that I mentioned, of course there are many advantages and disadvantages of using an one-step and a two-step RT-PCR. So let's know them one by one. Okay, so one step, the PCR process has many advantages. No, they give consistent results. The protocol is simple and fast. There is less pipetting because it's only one step. This technique is, however, less sensitive, and it's not possible to optimize reactions individually. It also does not provide stock cDNA. No, so one advantage of one-step PCR as well is the lower probability of contamination, because the sample is less exposed to the environment in a single tube, and the process requires less pipetting, as mentioned. Also, the results have higher reproducibility, and the procedure takes much less time. However, again, the limitation is it only uses one specific sequence-specific primer, and it needs fresh RNA samples to analyze true targets or repeat experiments, because it's very difficult to troubleshoot one-step RT step, on RT. So, and it's usually less sensitive, as I mentioned. So, how about two-step PCR? Two-step PCRs are extremely flexible and allow better control of your experiment. This technique allows individual optimization of the complementary DNA synthesis and the PCR reaction. It's more sensitive compared to the one-step method. Because random primers are used, the two-step method often runs more efficiently, and it provides stock complementary DNA, no? So, and that allows for aliquoting and use in the multiple assays. The two-step method does take much longer to perform and of course involves more pipetting and significantly more pipetting. The two-step RT-PCR allows use of random primers or oligo-DT primers, and the resulting complementary DNA can be stored for long periods of time and used for multiple PCRs. The complementary DNA template, the NTPs, polymerase, and salts are transferred from the finished RT reaction to the amplifying reaction. And because of this, it increases the chance of contamination. PCR is very famous right now because the government has been setting up a lot of molecular biology facilities all throughout the country because of this pandemic that we have, the coronavirus disease, no? Also, the causative agent of this is what we call the SARS-CoV-2. So what is SARS-CoV-2, no? So this is the accepted name of this virus, the severe acute respiratory syndrome coronavirus 2, no? The first one was in 2003 in Asia, the first SARS outbreak, no? So in risk communication though, we try to avoid with the name SARS-CoV-2, especially in the public, because we want to allay the fear, no? Especially to the World Health Organization, they want to allay the fear involved with the initial SARS outbreak, no? So they just termed this the virus that caused COVID, no? So that is SARS-CoV-2. So SARS-CoV-2 is a positive stranded RNA virus that expresses its replication and transcription complex, including its RNA-dependent RNA polymerase or RDRP from a single large open reading frame referred to as your ORF1AB. And the ORF1AB, RDRP, the ENS that is mentioned there, the envelope, nucleocapsid, spike protein, are genes, specifically the genes of these, are the targets most frequently used for the detection of SARS-CoV-2, no? Right now, the RT-PCR remains to be the gold standard or the standard method in detecting the COVID-19 disease, no? There are many moves to add more tests like the viral cell cultures, no? However, the issue of infectivity remains to be there. So we don't want that idea, no? Because if it's imagine culturing viruses, aside from the fact that culturing viruses takes a long time, it's very taxative to the laboratory to do that, no? And of course, we're worried that when you grow that, you will be infecting a lot more people, no? So right now, the gold standard for this is your RT-PCR, and again, it detects the parts, no? Specifically the genes I've mentioned, no? The ENS, ORF1AB. Okay, so in the next slide, I'll show you the current accepted RT-PCR based detection, no? The available ones, no? From China, Spain, Korea, England, no? Korea and Germany. So these are the current PCR tests available for this disease. So, and then in the next slide, I'm going to show you a short video about the testing of COVID-19 using RT-PCR. So, please listen to this.
COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2. When a person is infected, the most common symptoms include fever, cough, and shortness of breath. To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx. Then it is rotated for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase. Then the newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation. To extract the viral RNA by using commercial kit, the sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing, and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulse vortexing and incubated at room temperature. Then the virus is lysed under the highly denaturing conditions provided by the lysis buffer. Once the sample is lysed, a purification procedure is carried out by using a spin column. The sample is loaded onto the spin column. Then a centrifugation is performed. This procedure is a solid phase extraction method, in which the stationary phase consists of a silica matrix. Under optimal salt and pH conditions, RNA molecules are bind to the silica gel membrane, and at the same time, protein and other contaminants are not retained. After centrifugation, the spin column is placed into a clean collection tube, and the filtrate is discarded. Then a wash buffer is added. The column is put in a centrifuge again, forcing the wash buffer through the membrane. This removes any remaining impurities from the membrane, leaving only the RNA bound to the silica gel. Once the sample is washed, the column is placed in a clean microcentrifuge tube, and an elution buffer is added. Then, a centrifugation is carried out, forcing the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column. And a purified RNA, which is free of protein, inhibitors, and other contaminants, is obtained. After the extraction of the viral RNA, the next step is the preparation of the reaction mixture for PCR amplification. In this step, a master mix is used, which is a premixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase. Finally, to complete this reaction mixture, the RNA template is added. The tube is mixed by pulse vortexing. Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time. Next, the plate is placed in a PCR machine, which is essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target sequences in the RDRP gene, the E gene and the N gene. The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides onto the 3' end of the primer, synthesizing DNA complementary of the viral RNA. The temperature and duration of this step depend on the primer, the target RNA, and the reverse transcriptase used. Next, an initial denaturation step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase, and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists of heating the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next step, the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to 3' direction. The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. In the next step, the reaction temperature is lowered, allowing annealing of the primers to each of the single-stranded DNA templates, and annealing of the Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence. In the extension step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity, proportional to the amount of amplicon synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle, therefore, PCR can be used to analyze extremely small amounts of sample. For the measurement of the fluorescence signal, a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter, and a charge-coupled device CCD camera are used. Filtered light from the lamp is reflected off mirror, passes through a condensing lens, and is focused into the center of each well. Then fluorescent light emitted from the wells reflects off the mirror, passes through an emission filter, and is detected by the CCD camera. In each PCR cycle, light from excited fluorophore can be detected by the CCD camera, which converts the light that it captures into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it occurs in real time.
So that's basically the RT-PCR in the context of the coronavirus disease or SARS-CoV-2 RNA. Okay, so I hope you learned a lot from this lecture video. So if you have any questions, you can email me at my usual email address or send me a message at my Twitter account, for